I hope everybody can see that screen. And as Luva mentioned, a lot of the talks today will talk about different organismal groups in Madagascar. I'm going to talk about parasites and argue that they're an interesting category of organisms to study and can provide some interesting insight into some of the questions that began with Alexander von Humboldt more than 200 years ago. And then at the end, I'll ask for questions. So Humboldt was mainly interested in understanding the spatial distribution, or how groups of organisms differed across different geographical features. And this picture, which many of you may have seen and is fairly famous, here the group of organisms that he's interested in is plants, and the geographic feature he's interested in is volcanoes. In this case, this is the Chimborazo volcano in Ecuador. And in small writing, that is probably too small for those of you who are viewing today to see, he's listed the different plants that are in different portions of this mountain as you go up and down it, and this extinct volcano. And then on either side of this drawing, in these columns in German, what I think is he's doing is he's listing the features or the factors or the potential drivers, such as altitude or temperature or soil substrate, that may explain the different patterns in the plants that he's observed. Another geographic feature that Humboldt focused on was islands. In this case, this is the island of Tenerife and the Canary Islands. And again, he's showing how plants differ across parts of this island. And there's different plants found lower, closer to the coast, some higher up, and some in different sides of the island and things like that. But today what I want to do is replace plants with parasites as the group or the category of organisms of focus, and then replace the island of focus with Madagascar. And parasites are an interesting and highly diverse group of organisms, and Madagascar is an interesting and highly diverse locale. And so combining the two should provide some opportunities to observe interesting patterns. And that's the overall thesis, or the simple hypothesis I'm trying to develop in this brief presentation today. And so the objective is to outline the potential opportunities and some of the challenges, some of those challenges that we're able to overcome with the advent of new molecular methods, such as barcoding and genotyping, to study parasite taxa and continue Alexander von Humboldt's legacy of understanding how organisms differ across space and the patterns that describe, or the drivers that describe those patterns. First, I wanted to talk about some of our work that we've done using a human parasite, Plasmodium falciparum, a malaria parasite, and humans in northeastern Madagascar, where we'll use a model where hosts are sort of an island and parasites are colonizing that host/island, and how that model and thinking about hosts as something that is a suitable habitat for parasites to colonize can provide an interesting starting point to understand more about a parasite that we care about from a human health perspective. Next, I wanted to highlight some of the literature and other work that's going on that's showing that there's an emerging picture of the parasite diversity in Madagascar will match or is on par with the already well-known and very vibrant host biodiversity. Or in other words, I'll argue that understanding the host diversity of Madagascar alone is insufficient. And there's some new cool studies that are again leveraging molecular techniques and are creating a growing pattern of a rich parasite community in this well-known biodiversity hotspot. And then last, I sort of wanted to look ahead to the future and how we can continue probing the questions that were first posited by Alexander von Humboldt more than 200 years ago, and think about how we can go from parasite discovery or identifying parasites to describing how they're distributed and what is driving those patterns. And that'll have some clear relevance for human health, as disease emergence events that have happened in 2020 have already made clear, but also interesting implications for ecology writ large. So returning to that first item, this is our work about malaria parasites in northeastern Madagascar. And again, we're used to this biogeographical model of animals and plants colonizing islands, and it's fairly simple to apply the same model or the same scheme to thinking about parasites. In this case, it's a vector-borne parasite. So the mosquito is bringing a parasite that then colonizes a host. And importantly, those hosts remain in the environment and can be colonized and recolonized and multiply infected. And this is work that was done in collaboration with Maheri and many researchers, including Hedve Ranjayamadi, who should be on this call. So I hope it do justice. And here in this project, we're sampling human communities that live in the Makira and Mashwala forest areas in northeastern Madagascar. And there we have hosts. These are men, women, and children on the hosts that live in these areas. And we're gonna utilize some new molecular techniques, such as DNA barcoding, that allow us to track individual parasites or individual parasite genotypes. And that can allow us to categorize hosts into 3 types of hosts. There's uninfected hosts that I'm here showing in gray on the left. Then there's hosts that have a single parasite infection, and this could be with different parasites, for example, orange and green are different colors, showing that there's different parasites infecting hosts. And then there's a 3rd category of hosts. These are hosts that are multiply infected. And I'm showing this with a combination of colors with, for example, a host that has the green and the yellow colors combined has in a simultaneous co-infection of the green parasite strain and the yellow parasite strain. And again, the colors are arbitrary. But the simple hypothesis is that more hosts with multiple parasite infections mean that the parasites are more rapidly colonizing hosts, which indicates a higher parasite transmission rate. And if our goal is to eliminate malaria from human hosts in this part of Madagascar, from a public health perspective, observing many hosts with multiple parasite infections indicates that we need to do more control efforts or change the control efforts that are being used to reduce malaria sickness in this area. So if we return to the map, what we observe, as I'm showing here, is that hosts are often infected, and they're often infected with multiple parasites at the same time. So if this host is an island, it's getting colonized and recolonized. And in fact, I think it was 68% of the hosts contain multiple parasite genotypes at the time of sampling. In other words, humans are infected with multiple Plasmodium falciparum genotypes simultaneously, with a clear implication that we need better or novel control methods, especially in northeastern Madagascar around the Maro and Setra area and the Mashwala forest. So the sort of second thing I want to talk about is to quickly highlight studies that are emerging now that utilize molecular techniques to understand the parasites infecting non-human hosts. For example, lemurs, there's many studies coming out. And for example, they use molecular techniques to create a more robust phylogeny of this group of parasites. Again, this is the malaria parasites. But globally, we get a better understanding of how parasites and hosts have co-evolved when we apply those techniques to the parasites of lemurs, which are highlighted here. And that may be small to see. The main takeaway is that by studying hosts from Madagascar and applying new molecular methods, we get a better understanding of how parasites are distributed and why. In addition to mammals, there's also some studies on the parasites of reptiles, including a study of the blood parasites of chameleons. And here, there's a nucleated blood cells of chameleons, and next to those, there are some parasites that you can see in those cells. And just to conclude and wrap up, I just wanted to say that with these new tools, we're able to rapidly discover parasites and observe where they are. And it's time to transition towards understanding what are the ecological drivers of those distributions that we're observing. And molecular methods play a key role in that and allow us to extend the legacy of Humboldt and better understand the drivers of not just host diversity, but also parasite diversity. And that'll clearly have implications for wildlife health, ecosystem services, and human health. And with that, I just wanted to say thank you again to the organizers. I'm looking forward to the talks, and I will take any questions. Feel free to put the questions in the chat.